So I want to start today by telling you a story about 4 siblings, 4 sisters. And I've got a young daughter at the moment, so this is my standard mode of communication. And these 4 sisters, they were born off the coast of Queensland on a tiny set of rocks known collectively as egg rock. And they're so tiny that you have to basically zoom in 3 times off the east coast of Australia to even see them. Then we have the GBR on your left. And we've got the southern GBR here. And if you zoom in more to the Keppels, you see on the far eastern side a tiny speck of rock called egg rock. Now these 4 sisters were born a couple of years ago. And as every young Australian knows, it's pretty hard these days to try and find a house, a home for yourself, if you don't have support from your parents. But these sisters, they're square-tailed coral gropers, and their parents don't help them out almost at all. They're basically flung out into the open ocean as fertilized eggs, and they're left to the vagaries of the tide and the winds and the currents. But this is a happy story. This is a story with a good ending because all 4 of these sisters found somewhere to live. And that's a surprising story. 2 of them went westward from Egg Rock to Halfway Island. Ooh, that's inside my shirt. All right. So 2 of them went west to Halfway Island. All right. And they settled about 100 meters from each other. And that's a surprisingly long journey, because these things are only about 2 millimeters across. It's like you and I grabbing hands and then floating and swimming all the way to New Caledonia. So this is a surprising journey. It's a great journey, but almost as surprising as this story is the fact that we know this, that this story is a true story, that these 2 sisters did start at Egg Rock and they did end up in Halfway Island. Because if you went back 20 years and you asked the same question, the answer would have been, we don't know. And we don't know how we would know. 20 years ago, we could publish in one of the leading journals in the field an article which asked, honestly and defensively, are reef fish populations entirely open or entirely closed? So we knew that every year, adult fish went to the outside of the reef and they spawned eggs into the open ocean. And we knew that every year, larvae were coming in, they were settling and they were replenishing populations. But we didn't know if those settling populations were coming from the far-flung reaches of the GBR or whether they were the same eggs that we saw disappear just weeks ago. And in the interim, in the years since then, a bunch of researchers from this centre really led the way in developing a set of techniques that allow people to measure dispersal events directly. And in doing so, they really revolutionised reef fish ecology and particularly dispersal ecology. And that's Jeff Jones and Glenn Almany, you know the people, Hugo Harrison's talking next, Dave Williamson, Gary Russ, many others. They pioneered a set of techniques that could measure dispersal events in a concrete way for the first time. And then they got together the money and they had the audacity to roll these things out at enormous spatial scales to show that at management and ecologically relevant scales, dispersal was happening and what those patterns look like. And that is mind-blowing. And one of the most powerful techniques they developed was called genetic parentage analysis. You basically go to a whole suite of reefs and you sample juveniles and adults. You get DNA profiles of all those individuals, and then you cross-reference them looking for parent-offspring pairs. Because if you can find where a parent is, and you can find where its offspring is, then you know that in between is a dispersal pathway. And the genetic parentage dataset that forms the basis of this story is down there in the southern GBR. It's square-tailed coral gropers, and it's one of the most expensive and intensive and extensive parentage datasets that we have available. And it's answered all sorts of interesting questions for us. And to sort of express how important it is that we know what dispersal is happening, as a simple example, let's think of the dispersal of these juveniles from Egg Rock to Halfway Island, because it's an example of a particularly important and in some ways a particularly sacrificial form of dispersal. Because Egg Rock is protected, it's a marine protected area, and Halfway Island's not, it's fished. And for a long time, one of the centerpieces, one of the cornerstones, one of the pillars of marine conservation, particularly coastal conservation, particularly coral reef conservation, has been the designation of marine protected areas. And the argument's gone like this. Okay, look, we're going to take some of the areas you used to fish and we're going to put them in no-take zones. And that's going to mean a loss of areas that you previously liked. Now, that's a cost, but over time, the populations of fish in those areas are going to grow. They're going to get more numerous, they're going to get larger, and we know that's happening. We can see that, it's being documented extensively. But then, those more numerous, larger fish are going to produce many more eggs. And they're going to be dispersed out into the ocean, they're going to float around, and they're going to land on the areas that are still available for you, the fishes, to access. And there'll be so much more recruitment, there'll be so much more growth in the populations you're still able to fish on, that's going to compensate, it may even overcompensate for the area we took off you. That's larval subsidy effect. We've been selling that for 30 or 40 years. And until 2012, until a paper led by Hugo Harrison was published, there was never any real concrete evidence that was occurring. And they observed dispersal from green zones to blue zones, and everyone breathed a sigh of relief internally. So this data set that's giving us this information is much broader than just the Keppels, much wider than just egg rock. It was done over a whole swathe of inshore and midshore reefs, about 300 or 400 kilometers north and south around Gladstone. It was for two species that are sort of central to the recreational and commercial fisheries in the region, Plectropomus leopardus and maculatus. And it showed that this area was strongly connected at a scale that we hadn't previously suspected, that there was larval dispersal ongoing over hundreds of kilometers of reef area, and that the whole system was closely connected. And so we come to the last two individuals in our four-sister story, and they traveled north and they traveled east, and they went a lot further than the first two siblings. One of them went out offshore towards the Capricorn Bunker Group, traveled about 50 kilometers and was found near Heron Island. And the other undertook a much, much longer voyage, all the way north, about 200 kilometers. And it ended up in the Perseas, which is a hell of a long way to go. The dataset also showed that there was dispersal going in the opposite direction, and that all these different regions were connected to each other, and that there were also strong connections within the regions. Now, genetic parentage data doesn't really give us these trajectories. It tells us where they start and where they end, but it doesn't tell us what the route is. So what you're seeing here with this animation is a guess, right? But it's not just any guess, it's an incredibly sophisticated and complicated guess that's based on what we call a biophysical model. So, a biophysical model is like a chimera. It's like a hybrid of two different models. In its foundation, you have a model of oceanographic flows, and this one was developed by Lou Mason, who's an extremely good modeler of flows on the Queensland Shelf. And it's based obviously on the iron laws of geophysical fluid dynamics, but it's also based on high resolution parameterizations of the ocean floor. It's based on a network of arrays and sensors that tell us what's actually happening out there. But it's still a guess. Now, sitting on top of this oceanographic model, bolted onto it, is an individual-based larval dispersal model. And this one in particular was developed by Jeff Lees from the Australian Museum. And it was developed for these species, particularly for Plectropomus maculatus in this case. So it describes for each of these little particles what's going on in the larvae's head and what's going on in the larvae's body. So it decides how long it's been dispersing for, what its developmental stage is, how fast it can swim, what it can sense, and whether it decides to go towards it. All sorts of assumptions are built into it, but it describes, as best we understand it, the behavior and the decisions of individual larvae. And coupled together, these things will predict for us over vast space of sea where larvae come from and where they go to. And if we're very careful, we can match them precisely to the genetic parentage datasets. We can model the release of those two first siblings who went from, you can see them there in color, who went from egg rock to halfway island. And we can also simulate, because of the wonderful computational powers available to us, the dispersal of every other larvae that was released on that day in that area, and every other larvae that was released by that same mother. So not just 4 siblings, but 40,000 siblings. And you can see two things from this dispersal model. The first is that obviously to get to the same location from the same starting point, you don't necessarily have to travel the same routes. But that's just a little bit interesting. But what you can also see is that larval dispersal is much bigger and much more complicated than we can really see from a genetic parentage data set. And that's a fundamental limitation of genetic parentage that we have to get around. Parentage gives us the iron truth about what's going on in these systems, but only a very small subsample of it. And there are lots of questions in marine conservation, in coral reef conservation, that depend on a much more fulsome description of dispersal. We can't just talk about a subset of areas. We have to extend it over vast sections of the Great Barrier Reef, for example. And we can't afford to, and we don't really hope to be able to sample data on that sort of scale. And so the solution might be, okay, well, let's just use these biophysical models. Let's just, instead of sampling the data, let's just simulate it, because these models are very complicated, but the problem is we don't really trust them. The problem is they're built full of assumptions, and there isn't even really one model to trust. So, for example, let's say we said, okay, the spawning event happening here at Middle Reef might be occurring 2 or 3 days before the full moon, or it might be occurring 2 or 3 days after the full moon. And so we have a pretty good idea of when these individuals spawned, but not at that level of sophistication. So it's reasonable to suspect that we might be wrong by a day or two about when they left. Equally, we might be wrong about how big their sensory zone is, or the distribution of precompetent periods that they go through. We know they have different distributions through the water column over time, maybe to take advantage of bottom layers that don't move so much, maybe to avoid predation, maybe to feed, who knows. We have to assume things about that, and we're pretty sure that our assumptions aren't 100% correct. And so we don't really have a single prediction about what happens to larval dispersal. Instead, we have a whole bunch of them. We have thousands of them, in fact. And here's an example of our situation. Now, these are connectivity matrices. They're a way of visualizing in a sort of fairly straightforward way the flow of larvae throughout an entire system. Now, every one of these rows is a departure reef, so it's where eggs are leaving from, and every column is an arrival reef, it's where they're going to. And in the center there, you can see the parentage data set. That's what we sampled, right? That's the observed dispersal events that were reconstructed from the genetic analysis. Now, only a subset of the reefs were sampled, and so we're not going to see all the connections. And the samples that we took were not exhaustive, and so we're not 100% sure that those are the precise relative strengths of the connections, but those are definitely connections that are occurring. Now, arrayed around that genetic parentage connectivity matrix is at least, 5 different potential connectivity matrices, 5 different biophysical models with different assumptions that make slightly different predictions about how connectivity works. And if you blur your eyes, imagine there are like 1,000 of these damn things. And they're all slightly different, but they're different in important ways. The fact that the differences are subtle doesn't mean they wouldn't recommend different decisions be made. They wouldn't support different marine protected area networks. They wouldn't talk about different spatial harvest policies. And so how are we going to get around the fact that our data is either true but incomplete, or complete but possibly untrue? And of course, there's no element of suspense here. We jam them together into one big model. And we use the strength of the genetic parentage data to anchor the biophysical models into reality. And the hope is that if we start with a massive big ensemble of models, and imagine that those connectivity matrices arrayed at the top of this figure are all the various different candidate biophysical models we have to think about, we have to tie them down. We have to exclude some that are completely incorrect, and we have to worry about the remaining ones, which are fairly correct, which are probably not correct. We have to think about integrating them. Now, it might seem as though the genetic parentage data is a little too limited to sort through thousands upon thousands of candidate models, but it turns out that it's actually more powerful than I at first thought, and that's for the last time because of those 4 siblings. So imagine again, these are all of our models, and they all make predictions about dispersal in the system, and we ask them, because they're all modeled for the correct time, can you predict me 2 individuals moving from Egg Rock to Halfway Island? They can all do that. It's not a particularly long dispersal distance, and there are lots and lots of larvae being released, and so, okay, fair enough, we can handle that. None of these models are excluded or even really down-weighted on the basis of that particular question. And then maybe we ask for maybe the harder types of dispersal that we see, the longer distance dispersal. Can this model recreate dispersing larvae from Egg Rock up to the Percy Islands? Well, it turns out that most of them can do that too. I mean, a couple of them can't. A couple of them that don't assume very strong swimming abilities in the fish, for example, or might have relatively short distributions of precompetent periods, they have a bit of trouble in these long-distance dispersals. But again, most of them can handle a long-distance dispersal north, or maybe a long-distance dispersal out to the Capricorn Bunker Group. But these are part of a family. And what we're really asking is for these models to predict from the same release event 4 different dispersal events that go in diametrically opposite directions, that go long distances and yet retain the characteristics of strong self-recruitment in a regional basis. And there aren't many models that can do all those things at once when it comes down to these parentage data sets. And so we have an ability to really cut down the number of models we have to consider. Now, we don't get rid of all of them, and we don't leave ourselves with one true model. And that's reasonable. We'd expect that, right? Because we don't actually believe the true model is in this data set of simulations. And we know from other examples of modeling complex systems with relatively limited data, like meteorology, that when faced with a set of models, none of which contains the absolute truth, the best way forward, the most accurate predictions and the most robust predictions come from taking an ensemble approach to it, to leaving ourselves with a weighted subset of the original model set, which we use as a block to make predictions, and we think about the uncertainty within that block, and we use that as part of our predictions too. And so this, in some ways, I think is the future of larval dispersal ecology, of modeling the dynamics, the demographic consequences of larval dispersal. And we're almost finished, and I'd like to end it there because of thinking up with my time, but I guess I'd like to thank not just all the collaborators who made this possible, all the JCU researchers who contributed to this, Jeff Jones, Hugo, Glenn, Gary, and also to the institutions and the organizations that supported it. Thanks.